presentations. Each of us are going to have about 15 minutes or so for our presentation. And if time allows, we'll do a couple questions at the end of each presentation, but save most of them, please, for the end where we'll have a panel discussion. And then, I don't know, Josh, anything to add, or maybe introduce yourself, or you're starting off?
Yeah. So
you want to do the podium? Probably.
I'm going to head to the podium and do the first talk. And so I'll be starting off talking about some platform technologies to support 3D bioprinting. And this is work that's being done by RegenMed Development Organization. It's a nonprofit organization that is advancing a tunable bioink system. And that's what I'll be covering today. But we thought this would be a good talk to sort of set the stage for the rest of this session. So there's a variety of bioprinting modalities that exist, and shown here are inkjet, extrusion, stereolithography, and laser-induced forward transfer. And each of these bioprinting systems consists of three components: a cellular component, biomaterials, and printing modality and methodology. And so what I'll be covering in these next slides is a tunable bioink system that will allow for cross-compatibility with these different 3D bioprinting systems. And so why develop a modular tunable bioink system or a universal bioink? There's many reasons, but here are about 5 just to get started. So the cost of bioprinting hardware has come down significantly, and any lab can own and operate a bioprinter now. With widespread hardware, researchers often turn to their favorite biomaterials without much thought to printer compatibility. There's also been very little work to develop new materials specifically for bioprinting. No standards exist for guiding development of bioprinted tissue products, and there's no out-of-the-box compatibility between current biomaterials and bioprinting hardware. So essentially, you have to do customization for every case. So universal bioink could be tailored to potentially fit any device and any tissue type using a modular strategy. And so this Advanced Biomanufacturing Initiative, the REMDU Universal Bioink Program, is advancing a universal bioink with tunable mechanical properties for RegenMed additive manufacturing for clinical products. And shown here are milestones achieved over the first 5 years. Right now it's in year 1 of this program. So determine elastic moduli and tissue-specific parameters and generate formulations of a standard bioink. Create, characterize, and formulate bioprinted tissue constructs. Year 3, determine and optimize bioprinting speed and resolution. Year 4, generate optimal formulations for extrusion bioprinting. And then year 5, generate optimal formulations for inkjet and laser bioprinting. And so this universal bioink program consists of multiple stakeholders. We have industry partners that support this program with cost share, giving their time, maybe materials, equipment. We also have government partners, collaborations in advancing this platform technology. And what industry partners are getting out of this is one, knowledge base as we advance this tunable bioink system in areas of 3D bioprinting, and then also in developing standards. But this platform technology can also be used to develop individual products off of that industry sees benefit. And then, of course, the government's interested in advancing RegenMed technologies that are based off of this tunable bioink system. So the overall technical strategy consists of 3 main attributes. So there's the Satrapy, which essentially enables bioprinting control and compatibility for this bioink. There's biochemical customization, And what this allows is for tissue-specific bioinks to be created. And then another attribute is biomechanical customization, which allows for elastic moduli to be achieved to mimic specific tissue types. And so the first attribute that I'll cover is thixotropy. And essentially this property is where a material, when it's placed under stress, fails, which allows it to then be bioprinted. And then when that stress is removed, which is shown in this shear stress test here, the material then can retain a three-dimensional structure. And our old system was based on cross-linking of covalent bonds, but we're developing new technology that allow for this transient or reversible cross-links and to make use of this thixotropy property, which is quite useful in bioprinting. So one method that we're using for this thixotropy are catecholamine compounds. And interestingly, mollusks secrete these compounds, and that allows them to be able to stick to ship holes and rock surfaces and things like that. So initially, we were exploring this as a way to enable an adhesive hydrogel. And through further experimentation, we saw that there was also internal adhesion, which allowed for this thixotropy property to be exploited. And so this is one technology that we're using to incorporate into these hydrogels for the thixotropy. This slide is shown here that illustrates we have access to multiple different bioprinting systems, and we're in the process of testing the Bioink systems in each one of these bioprinters to ensure that there is cross-compatibility. And that's one of the milestones, is that we want to have a system that is essentially out of the box, and we can provide custom protocols for each one of these systems using our Bioink system. Shown here are some 3D constructs examples. So the upper left panel illustrates some of the internal channels that we can create in these hydrogels. The upper right panel is illustrating more complex patterning. The lower left shows that these bioinks are self-supporting, so we can layer on top. And then the bottom right illustrates that from a CAD image that's illustrating 3 channels, we can recreate that using our Bioink with fairly good resolution. That's the second attribute that I'll just briefly mention is biochemical customization. And so the example that I'm showing here is incorporation of laminin and fibronectin into our hydrogel is able to influence cell phenotypes. And so that image there illustrates when you just have laminin in the hydrogel, the cells take on a spherical phenotype. Fibronectin, they're more spindle-like. And then there's an intermediate phenotype when you're using both laminin and fibronectin incorporated into the hydrogel. And taking this a step further to actually look at function in 3D liver constructs or organoids, we see that with the incorporation of fibronectin into the hydrogel, this actually improves both these organoids' function with the production of albumin and their viability. And then the left panel is just showing that we can create more complex shapes using this tunable system. Taking this a step further, we're also able to evaluate FDA-recalled drugs using these 3D organoid systems, and they show appropriate responses. So in summary, We're working on a tunable Bioink system for 3D bioprinting. I talked about 3 attributes of this system: the Satrapy, the biochemical customization, and then the biomechanical customization. Long term, we believe that this platform technology will bring down some of the barriers in tissue product development by enabling cross-platform compatibility, for instance, also with developing standards for bioprinting and bioink development. And the latter slides, or the latter talks today, we'll talk about some of the applications of 3D bioprinting, and this bioink system will help enable such things as tissue engineering and organoids for drug screening. And I'll end with an acknowledgement slides. So for the RemDo Bioink team, the lead technical person is Dr. Alex Scardo from Wake Forest. And we also have another academic collaborator from Rice, and that's being led by Dr. Tony Mikos. So with that, thank you. And I did want to make one other announcement. We're also launching a new professional society, the Regenerative Medicine Manufacturing Society, and we'll have the inaugural launch meeting this Friday here at the World Stem Cell Summit. And it's a forum for sharing the latest developments in the field, providing scale-up of RegenMed manufacturing technologies. I have more flyers up here if you'd like, and you can speak with me afterwards. And that's it for me. Thanks. What time? It starts at 8 and runs till about 4:15. It's also, if you have that handy exhibit guide, somebody has that handy. If you have this handy exhibit guide, it's also in there. The agenda has changed slightly. We've added a few additional speakers, but for the most part, you can look in there, January 26th, page 15, and it'll give the different sessions. So I'm going to turn the stage over to
Do you want to take a few questions if there are any?
I can take a few questions if there are any on this tunable bioink system.
If not, we can do it all on the panel. Okay. Go ahead.
So next up is Dr. Tom Shupe. And he's from Wake Forest University, and he'll be talking about capturing the cellular microenvironment.
Thank you all for the opportunity to speak. As Josh said, I'm an assistant professor at the Wake Forest Institute for Regenerative Medicine, and I've been the scientific director for a program that we've been running for a while. We call it the ECHO program. It stands for Ex Vivo Console of Human Organoids. And what I was asked to talk a little bit about today is what is the integration of bioprinting and bioinks in the ECHO program. So these aren't bioprinting individual cells. These are bioprinting tissue organoid constructs that have been fabricated prior to bioprinting. The specific aims of the ECHO program, we were tasked to create an integrated human organoid construct system. And by integrated, I mean different tissue organoid types are maintained under a common media. They communicate with each other. The media is recirculated, and it allows us to do integrated pharmacology and toxicology studies where a compound is perhaps metabolized by a liver organoid and exerts an effect on a downstream, brain, testes, cardiac, and through the entire program, we've maintained compatibility of the organoids with the bioprinting process, and I'll show you on the next slide why we want to do that. And the goal of the program was to come up with a system that was comprised entirely of human primary cells, commercially available primary cells, and sort of get an integrated system where we could test the effects of drugs and toxic agents. And what we use bioprinting for in the ECHO program is the application of the tissue organoids into some different geometry microreactors that we've developed. And the idea being that if we want to scale this up and mass produce these for perhaps drug discovery or pharma applications, we would have a Bioink and a bioprinting system that we could rapidly apply the tissue organoids to microreactors in a precise and reproducible and scalable manner. And most of the tissue organoids that we use or have developed are spheroids, and you know, they're largely internally supported by some of the strategies we've used to fabricate the organoids, and we'll be talking about that actually at 4:15 today, the tissue organoids themselves. But a large percentage of the cells in the tissue organoid are actually on the surface of the organoid, and we've designed bioinks that are supportive of those cells that are on the surface and aren't completely surrounded by their neighboring cells. So that's sort of the thought behind why we put effort into customizing the bioinks. We've known for a long time that, you know, traditional 2D cell culture fails to recapitulate a lot of the normal components of the cellular microenvironment. And in our tissue organoid design strategy and in the supportive hydrogel matrix or the bioink that they're embedded in, we've tried to capture as many of these missing components as possible, and that's, you know, given us the long-term viability and function that we've seen in our system. And for these components, cell-cell contact, growth factor concentration near the surface of the cells, cell matrix adhesion phenomena, and the chemical and physical properties of the surrounding matrix. These are properties that are directly affected by our bioinks. So this is a slide actually from the tissue organoid lecture that I'll be giving later, but what we found is that the largest contributors to maintenance of cell phenotype and viability in terms of microenvironmental properties are the biochemical properties, the mechanical properties, the cellular properties, or the cellular composition, and the cellular microarchitecture. And the biochemical and mechanical properties are dictated, at least to some degree, by the matrix that the cells are in contact with. So the first key component of our bioinks is they contain tissue-specific decellularized matrix, extracellular matrix. So our liver organoids are surrounded by hydrogels that contain solubilized liver extracellular matrix. Our cardiac organoids are surrounded by hydrogels that contain decellularized cardiac matrix. We've been decellularizing tissues for close to a decade now at WFirm, and we find that a lot of the structural proteins in the matrix survive the decellularization process. Fortunately, the polysaccharides associated with that matrix also are preserved throughout the decellularization process. This is an alcium blue stain, which shows polysaccharides, most of which are glycosaminoglycans. Glycosaminoglycans are polysaccharides on that decorate extracellular matrix structural proteins and are able to bind growth factors. The reason we want these in the matrix is because, or in the hydrogels, is we want growth factors that are either added to the system or autocrine factors or paracrine factors that are produced within and around the organoids. We want them concentrated near the cell surface where they can react and associate with the receptors. And having these glycosaminoglycans that are able to bind signaling molecules and bioregulatory molecules that holds those components near the cell surface where they can act. So for our solubilization, we decell organs, take that organ, lyophilize, digested trypsin and pepsin and hydrochloric acid, and come up with an extract, and that's added to our bioinks. And when Alex Skardal did a lot of this work, when he suggested that we digest these matrices, I thought we'd destroy any biochemical function that they might have, but it in fact worked out pretty well. I'll show you one thing. We initially, I don't know if can you see the red in the lower left? Initially, we were lyophilizing and cryomilling the matrix and adding that powder into the hydrogels, and it killed the cells. And these are hepatocytes. Hepatocytes can be phagocytic. What was happening was those particles were being phagocytized into the cells, and they were clogging up endocytic vesicles, giving the cells indigestion, and they were dying. With the solubilized matrix on the right, and these are human primary hepatocytes at 7 days, we maintain almost 100% viability with the solubilized matrix and a hyaluronic acid hydrogel. Here's some more evidence that these hydrogels with the matrix in are supportive long term. These are 4-week cultures, and you can see on the left in a collagen sandwich gel, you lose basically all viability and function. With the hyaluronic acid hydrogel supplemented with liver extracellular matrix, we preserve viability and function out to 4 weeks, and you can see we have increased functional activity by the cells in terms of metabolism of compounds. The lower right-hand panel is showing that, and this is a LEE is the extracellular matrix extract. The TE is the tissue extract, whole tissue extract. You can see we actually have good retention of a lot of key growth factors in the solubilized matrix, and even better retention than you see in whole tissue. Here's some more evidence that the extracellular matrix that is added to our bioinks is binding and concentrating growth factors. This is hepatocyte growth factor, which binds very closely with components of the extracellular matrix. And you can see that if we take this matrix and incubate it at increasing concentrations of HGF, and then wash until we don't lose any more HGF, you can see that it does bind the growth factor and doesn't let it go. We repeated this with EGF, that it's more of an electrostatic interaction with the matrix and doesn't stick as well. And you get a similar curve, but then as you wash, it washes out at about the same rate. But this shows that the matrix does bind and hold growth factor. Here's some viability data and some morphology. This is 7-day primary hepatocyte culture. On the right, you can see we've got the cells on a hydrogel containing extracellular matrix, and we get good preservation of viability and morphology. One of the main ways that we've found that the ECM changes the cells and seems to make them behave more normally is E-cadherin is a component of a cell-cell matrix, or a cell-cell adhesion complex. And you can see that the E-cadherin is expressed to a much greater degree in cultures that have the solubilized extracellular matrix. And to some degree also, cell matrix interactions are increased, as you can see by the increase in beta-1 integrin expression. There's also a clear effect on hepatocyte cell function. This is normalized by DNA content, so this is like per live cell. And you can see we've got a great increase in albumin production in the presence of the solubilized ECM. The second property of the hydrogel bioinks that we have tried to take control over is the elastic modulus. Organs exist at a range of stiffnesses from blood all the way up to bone. And due to mechanosensing by the cells, in order to maintain appropriate phenotype, you have to get close to the normal stiffness of the organ that the cells are extracted from. And Josh alluded to this a little bit. This is a system, once again, developed by Alex Skardal at our institute. He's come up with a system for setting the stiffness of the hydrogel both before printing and after printing. So he uses a thiolated hyaluronic acid or gelatin, and then he uses 2 types of cross-linkers. He uses a polyethylene glycol diacrylate and also polyethylene glycol that's decorated with alkenes. And the PEG-DA acts as a cross-linker for the thiolated hyaluronic acid or gelatin. And that is a spontaneous cross-linking that occurs at any pH above 7. So you can set the initial stiffness of the hydrogel Bioink, and then and you can tune that stiffness to be appropriate for extrusion bioprinting or whatever you want to do with it. And then when you want to match the stiffness of the native organ after you've bioprinted, then you can use UV light to activate the alkene cross-linker, and that'll set the final stiffness. So by using different geometries and concentrations of these modified polyethylene glycols, we've been able to mimic the stiffness of a variety of organs up through muscle. So this has been a good system that we've been using to get a two-stage printable and then final stiffness to the hydrogels. Here's one more piece of evidence on how the stiffness of the hydrogel can affect phenotype. And if you look at the bottom, that's Phalloidin staining. So this is organization of the actin cytoskeleton. And these are, once again, human primary hepatocytes at 7 days. And you can see on the left that it's a more punctate staining that would be associated with a more modal phenotype. On the right, it's a more dispersed and diffuse staining across the cells, and this is representative of a more sessile phenotype. So there's direct impact on phenotype based on stiffness. And this is not directly affected by our bioinks, but the number one contributor to long-term stability and function is having cells in contact with the cells that they expect to be in contact with. So I'll talk about this a little bit more at 4:15, but all of our tissue organoids contain all the major cell types found in the native tissue. And we buy these independently from different primary cell vendors and titrate them together and form our tissue organoids. And but that spherical organoid is then embedded in a hydrogel that supports the cells on the outside, and you can get a good appreciation for how many, the large percentage of the cells that are in contact with that matrix from that picture. So with that, I'd like to thank everyone for their attention. This is the team that did all the work on the ECHO program. In terms of the bioinks, Alex is probably the number one contributor to that, but at W Firmware team, so we all pitched in a little bit. Thank you very much.
Okay, I'll be next here. My name is Keith Murphy. I guess I'll give a brief background. I'm going to talk about a little bit of a different perspective, an industrial perspective, and I'll go a little bit into detail on Organovo's technology, but also speak about the industry in terms of the pharma industry viewpoint on opportunities for these 3D models in drug discovery. My background quickly, I'm a chemical engineer by training, went directly into industry and worked at Alkermes and Amgen, both 10 years at Amgen, first in drug delivery, so sustained release of proteins for days from injectable microparticles. And then during my time at Amgen, went to business school at UCLA and then converted over to be a more team leadership, product and strategy team leadership, and led a team that developed, I was one of the team leaders for denosumab, which is sold as Prolia and Xgeva, so it's a monoclonal antibody for rank ligand inhibition that is marketed today, and it sells about $3.5 billion a year, so a big win for Amgen. I moved on from that and founded Organovo in 2007, so I'll talk a little bit about the Organovo story. Today, I run Viscient Biosciences, which is a company that's leveraging actively current 3D models in drug discovery and NASH liver discovery. So brief background on Organovo. Organovo has been around for quite a while now, went public in 2012, and has developed a number of tissues through a bioprinting technology, a core bioprinting technology that came out of the University of Missouri. Professor Gabor Forgac, who I'm sure many of you know from the University of Missouri, had the original technology that we licensed and built the company around. So Organovo is publicly traded, about 120 employees in San Diego now, and has built liver tissue, kidney tissue, a number of different tissues, has an active commercial offering, especially in the liver and kidney tissues, and especially in toxicology, so testing pharma drugs for their safety and efficacy, and there's quite a bit of data around that. I'll show you that, that data in brief as I go through here. So Organovo's business model is to leverage different tissues in a number of different verticals, and the verticals are shown on the right of this slide. In brief, there's product and services in preclinical safety, so tox testing, as I mentioned. And for that, you use a normal tissue, so you use a tissue that is healthy, made from healthy donor cells. We tend to use, or Organovo tends to use, mainly primary cells, primary human cells, and so you offer a normal tissue and test the drug against that. And then efficacy and disease modeling is the next step from that in an in vitro drug modeling setting, or disease modeling setting. And there you're actually sometimes using a normal tissue and inducing disease, but often you're using a diseased tissue that can be built using patient cells. So you can start the bioprint with a source that comes from patient donors, which involves a lot of work on the cell sourcing side and academic partnerships and things like this. Or you can induce, of course, a disease in the normal tissue, much like you would in an animal model. And then Organovo is highly focused now as well on therapeutic opportunities, so transplant tissues, which is of course near and dear to this, the broader World Stem Cell Summit, and has an active program with liver that I'll talk to you about a little bit briefly, just to set the stage on what Organovo's major focus is becoming. It is very focused on the commercial success of its launched businesses, but some of the major value for Organovo in the future is this therapeutic liver tissue. So this has advanced quite a bit from when we started Organovo, and it's a patch that's about the size of a dollar bill. Organovo's public messaging has said a dollar bill. I tend to think of it as a cell phone, a smaller cell phone, not a Galaxy Note or something like that would blow up inside you. But if you think about a cell phone, the thickness is similar to a little bit thinner maybe than a lot of the cell phones, but about 1 millimeter. What all the cell phone makers want it to be, about 1 millimeter thick, and about the size of a dollar bill or your cell phone. And the patch is intended to treat patients with a severe need, and these tend to be folks who have acute on chronic liver failure, so essentially transplant patients that need a liver transplant, but also a really interesting opportunity in inborn errors in metabolism that I'll talk about a little bit. And Organovo's advanced this to the point of extensive use in animals to date, and targeting an IND for this within 2 to 3 years. And has a lot of data emerging that shows this is working very well to date. So the rodent studies have showed excellent engraftment in the animals, and you can see with the arrows there's a large network of microvessels that form. There is an endothelial element to the patch that allows engraftment with the native host tissue. And you do see the formation of these microvascular networks, and then importantly, you can see from the staining here, production of human factors inside the tissue, but Organovo has also measured those circulating in the serum. So obviously you put a patch into an animal, if you measure human albumin circulating or human alpha-1 antitrypsin circulating, that's coming from the patch. And then most recently in October, Organovo announced its initial proof of efficacy in an animal model. So there's a long way to go. We regularly cure cancer in mice, and then it doesn't quite work out in humans when we take the drug forward. But it's a step that needs to be taken anyway, and Organovo was able to demonstrate alpha-1 antitrypsin cure in an animal model. So alpha-1 antitrypsin is a factor that is made in the liver. Many factors are produced by the cells of the liver that circulate in the body and are important for a number of biological functions. Alpha-1 antitrypsin is one of those. For people that aren't familiar with the biology of alpha-1 antitrypsin, the best comparison is actually clotting factors in hemophilia. Hemophiliacs have a genetic problem that it essentially expresses itself in the liver cells. So the failure of the genetic code is a failure to create these blood clotting factors in the liver. Hemophiliacs are well served. We're not going to go after that opportunity because they're so well served as patients already by available treatments, but alpha-1 antitrypsin works similarly, and there aren't good solutions. And what happens is over the course of time, these patients end up needing a liver transplant from a donor just to get enough factor of this alpha-1 antitrypsin in their system. So that's the kind of opportunity that Organovo is going after first. But I wanted to give you that background just to set the stage and to talk about the in vitro uses of the tissue and then specifically get into drug discovery as well. So the same liver tissue, but in a much smaller format, is used in Organovo's commercial offering in vitro tox and in disease modeling. And that tissue ends up being, as you can see here, in a 24-well plate if you look in the upper right. So the size of this is about 3 millimeters across by 0.5 millimeter thick, which is sufficiently in 3D enough to get the biological function that you need. And Organovo's base offering includes hepatocytes, stellates, and endothelial cells with a micro-patterned architecture that doesn't match the liver interior architecture, but has a lot of elements of it reproduced. And that allows you to get really good functionality. And there's a lot of data around this. We don't have time to go into all of that. That's not the intent of the talk. But to show you some of the, what Tom was discussing, the cadherin, you can see in green here, the E-cadherin staining, showing you're getting tight junctions forming in the cells. That's critical to normal cell function, and that's what is missing when you plate out hepatocytes. They fail to offer normal liver biology. Right away they start failing, and they fail to even make albumin after a couple of days. And so it really is a 3D context you need to put liver cells in to get good performance. And the sort of summary data of that is shown here, where if you compare 2D hepatocytes, which has been the gold standard for pharma for many years, you have poor conservation of liver enzymes, a few days of longevity of those in terms of expressing a liver cell phenotype and producing albumin in these things. You never get cholesterol biosynthesis, whereas in Organovo's tissue, all of those things are present, and they haven't really gone beyond 42 or more like 60 days, but presumably this tissue would persist past that time. It's ongoing, it's at steady state essentially at that time point. And then importantly, in terms of proving the function as well and the relevance for drug toxicity, there have been a number of drugs tested for comparison to clinical toxicity, which is really the key. And where toxicity prediction has failed over time, it's because of the combination of 2D hepatocytes and rodent models, which are the gold standard when you combine those things. If you don't see toxicity in that system and you take a drug forward, you still have about a 5% rate of failure. We did a pretty good review with an outside consultant of the rate of failure. So when you put a drug into phase 1, the likelihood that that drug is going to fail specifically due to liver toxicity as you take it forward is still 5% today, prior to the use of Organovo's platform or other emerging platforms. And so that's a pretty high number when you think about it, and the high cost when you think about the costs of running these studies in the hundreds of millions of dollars. And you don't see it in phase 1, you don't see it in phase 2, usually because you're using such small sample sets as well. And sometimes these things take more time to emerge because your early studies are usually short. So there's a number of things that won't be seen in the hepatocytes because of the poor function. They won't be seen in rat models because of species specificity of the result. And Organovo has predicted a number of those, or shown that the tissue expresses a number of those. So you see for troglitazone and trovafloxacin, which were two of the more dramatic stories of drugs that were withdrawn from the market, you see that, or failed in late-stage clinical trials, you see that the tissue predicts those, and importantly shows a nice pattern of methotrexate-induced fibrosis. Methotrexate is, of course, a drug that is used very regularly in a large number of patients, but it does have quite a high level of toxicity. Not as, or acetaminophen is another example of a drug that has toxicity, but we manage it clinically, right? So you don't give methotrexate to everybody because they would have liver failure, but you give it to patients because of the benefit, the huge benefit in certain cancer indications, in rheumatoid arthritis. It's used. But the nice results in this tissue also show that even though when a toxicity result is not binary, either it's massively toxic and you won't advance it, you can start to understand the toxicity of a drug. And then you can develop compounds that you might not otherwise, because you can be more predictive and you can be more confident that you can predict the toxicity. And if something can have a significant clinical benefit, then you manage to the toxicity. And that's certainly the case with methotrexate. These patients who go on methotrexate chronically, usually for rheumatoid arthritis, are monitored long-term because about 20% of them do develop liver fibrosis because of the drug. That's why a lot of the biologicals have had such success over time, because they're safer. And methotrexate is usually where you start with a rheumatoid arthritis patient. They will end up on something like Enbrel or Humira over time. And they're switching because their liver biopsy shows that they start to get, in many cases they switch. There's a number of reasons, but one is that you'll start to see the liver fibrosis form. So back to the context of using an in vitro model to predict this, the University of North Carolina, working with Organovo's platform, did methotrexate exposure studies and showed a very nice pattern of liver fibrosis is replicated in the tissue. And so this is a disease model then. As you can see here, there's a clinical biopsy sample in the smaller inset compared to the larger panel there. And what you see that's conserved is that you get pockets of fibrosis, and then you get what's called this bridging fibrosis across, so it's a long tendril of collagen or ECM across space. And that is the clinical pathology of methotrexate-induced fibrosis. And so you show a nice correlation between the clinical disease and the tissue. And this leads for opportunity in a number of ways, and specifically is one of the things that led to what I'm doing next, which is working with a company called, well, we started up a new company called Viscient Biosciences. Viscient is overall a company that will take advantage of these new drug discovery opportunities, these new models. So we believe broadly in the power of these models to be predictive, as I think, go back here, as I believe this was the first really strong, compelling case that started to bring pharma companies to Organovo rather than the other way around, where you're sort of pushing to get their attention, where you're saying, hey, we're actually replicating disease that's known, the clinical pathology of disease. That becomes very exciting. And so what Viscient is founded to do is to take advantage across the industry of 3D models that can be predictive of disease and do drug screening. So as I said, Viscient is essentially a pharma company at a very early stage. And we intend to be agnostic as to platform. So we're starting, because of my background and my relationship with the company, we're starting with Organovo's platform as our first interaction. And as I showed you earlier, that is part of Organovo's business model, to partner with pharma to do early studies on disease modeling. And that's what we're doing essentially, is contract research by Organovo, paid for by Viscient, to leverage this model and build a NASH fibrosis model. So not methotrexate-induced fibrosis, but NASH, which is a bigger commercial opportunity. And we're working hard on that together right now. So Viscient, then the other folks in Viscient come from Ardea Biosciences, which was so Viscient is also based in San Diego, like Organovo, so we've got proximity there to help move things forward faster. And Jeff Miner is the co-founder and chief scientific officer of Viscient. He comes from Ardea and a small team out of Ardea as well. Ardea was a company that developed successfully a number of drugs. The whole company sold to AstraZeneca. So pharma companies, as opposed to maybe cell therapy companies, which don't often do it, although now we're starting to see it with CAR T cells, often are bought out entirely by big pharma. That's what happened with Ardea. So they were successful in developing their drug, and the reward is, of course, that they have to go look for a job after that. Thanks for the molecules. Thanks for the commercial opportunity here. We'll take it and run with it. And now we're going to wind down the company over time, and that's very normal. Happens all the time in San Diego, and people end up in new efforts. That's exactly what's happening here at Viscient. And so Jeff brought along some of his team, and we're working together to do development specifically in NASH. As I mentioned, the first step in that is to leverage the Organovo platform that I described to you for studies in liver fibrosis, specifically in NASH, non-alcoholic steatohepatitis, which is fatty liver disease. Very large commercial opportunity and very much an area of interest for pharma. There are a number of other pharma customers that work with Organovo on this, but I think Viscient may be unique in the sense that we, as a company, are starting with the premise that these models will lead us somewhere, and others, I think, are more dipping their toes in the water at this point. But pharma, I think, is increasingly aware of the opportunity. So to kind of talk at the high level about how the industry sees 3D cell models, pharma is increasingly aware of the opportunity and is starting to build teams to specifically address this. So GSK, for example, has a head of complex in vitro systems with a small team who's targeted at helping individual disease model and drug discovery groups leverage these technologies to have some, they'll have some awareness of the space, some projects that they're doing, but it's all in service of the larger goal, which is to have individual disease teams who are doing discovery work with specific models. But I'd say pharma is still dipping their toes in. I don't think they're as broadly committed as they will be in the long term, partially because they don't spend a lot of money in drug discovery. That's one of the big challenges. It's an area, I mean, on tox, broadly speaking, when you add all that up, it becomes a very large number because every drug has that. But when you're talking about an individual molecule, like, for example, in NASH fibrosis, just a team working on that, they get a budget, but they don't get the budget that the clinical teams get. And so it's a small percentage of pharma spending overall, and then it leads and grows over time. So it's hard to get them to see them committing to as much of this as they really need to be successful. And a good example. So I tend to do my research as a having gone to business school. One of the things you're taught a lot about is case studies. And so starting up a pharma company for the first time, since my previous work was all more in technology. I read a lot of different business cases. There's 2 books out there on the formation and success of Vertex. If you've ever read those books, some in the audience may have. The $1 billion molecule, and then the follow-up, The Antidote. And the whole concept there was pulling the people out of pharma who basically were tired of the budget limitations within pharma and had the opportunity to do more and invest in new areas. Specifically in that case, Vertex was focused on rational drug design. Was the first company focused on that, but you need to fund it separately. They needed to do it outside of pharma to really make it successful. So that's why I see Viscient having an opportunity here. We're committed to the opportunity and to really growing what we're doing, and we're going to be partnering with academia in identifying opportunities and funding them where appropriate. And then the other challenge that I think everyone sees, the farmers that are working in this space and then ourselves as well, that's important for everyone to think about and for academia and the broader industry to help solve, is the trade-off between throughput and content. And this is the big challenge, right? So pharma works often in a high throughput screening context. If you're going to go find drugs, you need to start with a large pool of chemical compounds, find hits, and then move to the next stage and whittle that down to a drug candidate or a small number of drug candidates. And even when you look at the Organovo technology in a 24-well plate, and they've done work to move it towards 96 well plates with work with NCATS, the NIH Institute that tries to help throughput as well. But right now, 24 to 96 is the limit, and you're not going to screen 10,000 compounds at that scale, and you can't afford the cost of doing that. So there's this trade-off between everything we're doing as an industry to move into higher content assays, which are these rich 3D tissues or organoids that have a better biological result, but you trade off on the throughput. And so where we're going to have success as we focus longer term is finding the sweet spot and finding the maybe parallel things that can work together, where you've got one thing that's at higher throughput and one thing that's at higher content. You may not get as rich an answer from the higher throughput, but you need to start somewhere. And so that's one of the thoughts I wanted to put out there, and maybe we can have more discussion on that. But for now, I'll rest. So let me turn it over now. We're going to switch to the next presentation, and we'll have Dr. David Lott from the Mayo Clinic. He's an associate director at the Mayo Clinic come up and present.
Good afternoon, everybody. Thank you for coming to this session. So yeah, a little bit of background. I am the Associate Director for the Center for Regenerative Medicine at the Mayo Clinic in Arizona, and also have a lab that investigates tissue regeneration for head and neck, for the body of the head and neck. In addition to the kind of the head and neck regeneration side of things, the reason I first got into that is I'm a laryngeal surgeon by training. So I work a lot with, obviously, cancer patients and patients that have had various traumas, and trying to investigate how we can regenerate some of these tissues for reconstruction, because you can imagine for head and neck structures, having loss of function for those tissues can be very disabling for a lot of people. So our lab is really based at looking at that. So what my part of the talk is a little bit different than kind of what everybody else is looking for. Is, you know, in addition to 3D printing, bioprinting for kind of human organoids and kind of research systems, is how do we use 3D bioprinting for reconstruction? How do you regenerate tissue for failed organs? How do we use it for actually replacement of damaged tissues or at the time of cancers and things? So, and as a surgeon, I'm very interested in how do we get these things from the discovery phase into the translation phase as safely and as quickly as you possibly can, so we can increase the benefit for our patients with that. So, you know, 3D printing, I'll just go real quick, brief overview of that, because I'm sure you're all familiar with what 3D printing is at this point. But you can see that over just even the past few years, the number of publications in PubMed on 3D printing issues has just skyrocketed. And as a result, so has grant funding and relations in terms of with industry funding for different projects. So this is really where the future of a lot of medicine is going. And I think it's ripe for us to really grab a hold of it and make a lot of benefit for people. You know, there's a lot of good examples really already of things going through into the phase and how well bioprinting can replicate the natural structure and in some cases the natural function of the body tissue. So here's an example of our friends at Wake Forest using a 3D CT scan to 3D print an ear and have that obviously very similar shape to that. So at the risk of having a little bit of hubris with this, Mayo Clinic, we're much farther than everybody else is on the panel in terms of human organoid bioprinting. So we use the same model that Vicanti did to use the ear, and we can actually print our own human organoids on the back of that. So, yeah, so right. Maybe a little bit of exaggeration, but so what we're really looking at in terms of just the 3D printing process. So we discussed this a little bit also. So in 3D printing, there are many different ways that you can print a whatever structure you want. But when you're looking at 3D bioprinting, obviously the process has to be one in which is biocompatible. So you have to have it that cells can survive the process. Which takes away a lot of the traditional options. So what we see now really is there are some options with stereolithography, but really what we're seeing more is like the Bioink jet printing or kind of extrusion type printers that use air and different pressure actually to get to the biomaterials out. And where some of the work that we've been listening to today is, you know, how do you get these structures out, these materials out, without damaging them through that printing process? And so that's where a lot of that comes from. The benefits of 3D bioprinting, you know, like we've been talking about already, you know, you have the ability to print a combination of cells, growth factors, and biomaterials all together. So instead of having to do those separately and try to figure out how to put them together, theoretically, you can get the cells and growth factors and biomaterials all in one area, so it's kind of ready to go. You can get the cells deeper within the constructs. You can have the growth factors within the constructs. So there's a lot of options that are there. In terms of reconstruction from my side of things, because you can base these off of CT scans, you can have a patient-specific product ready to go for that patient versus trying to kind of do a cookie-cutter approach for everybody. You can get exactly what that patient needs, and I'll show you some examples of that. Once the process is in place, getting the 3D printer and the whole process there can be very expensive and very time-consuming. But once that process is in place, you have the ability to quickly and reliably reproduce these scaffolds and these structures that you've been working on. So from that standpoint, it can be very cost-effective, increase your productivity, and very reliable. You're printing the same structures over and over again with very little variability, so you know what you're going to get at the end of it. So again, it gives us improved reconstructive options. This is also seen by many as maybe where the future of transplantation is going. Because we don't need to have the immunosuppressant medications, this can maybe supplant some of the transplantation. And at this point, we really don't know what we don't know about bioprinting. So the possibilities are out there, and it just takes a lot of time and effort to figure out what those are. Some of the drawbacks, there are drawbacks to everything. At this point, it is a new technology, and there are a lot of unrealistic expectations out there, especially for where we are currently with bioprinting. And so we still need to do a lot of research to figure out really what the limitations and capabilities are. And then the other big limitation that we're going to talk about a little bit today is, the FDA really doesn't know what to do with this yet. And I'm sure you've heard that throughout this conference already. I've heard it a bunch of times. And trying to figure out how do you navigate that. And so what I wanted to do with the talk is use some of our work as an example on how we've been working hand in hand with the FDA through this process to kind of understand from each side what is needed. So that maybe then facilitated a discussion at the end of this on how we can use some of these technologies and techniques to actually get those into patients a little more quickly. So the first thing is, what the FDA is looking at, and we just had a meeting with them last week in trying to get these very things into patients. And they're not as much concerned with function, although obviously they want things to work, but really the priority is safety. And if we're going to put an implant in that's within the body, what is the strength of that implant? Is that going to be able to sustain the natural stresses and forces that are in the body throughout the lifetime of the person, whatever the expected life frame is going to be for your implant? If you say it's permanent, does that mean really permanent, or does it just degrade over 20 years? What does that mean? You have to be able to document what that degradation rate is going to be. And also looking at tissue integration. So there's really two things that we look at with bioprinting. Is one, how does that scaffolding and the cells and everything integrate into tissue? How do blood vessels grow into it? How does the tissue around that scaffold interact? Does it form a capsule, a protective capsule around it, or does it truly integrate into the body? And the other part of that is the cellular integration. So it may be one thing to have a scaffold that integrates into the body, but can the cells interact correctly with that scaffold, both on the outside and then actually once it's implanted into the body? So what does that cellular integration onto the scaffold really look like? Then again, obviously, what is the immune response? Does it wall it off as a protective shell, or does it integrate? You get small levels of immunosuppression around that. So, very small immune reactions can have significant consequences when you're talking about implanting these into cancer patients, because you don't know what that immune environment is going to be like at the time of surgery. And although we think, we know stem cells have an immunoprotective response to it, but really when it interacts with tumor cells, if there are residual tumor cells, what is that going to look like? And then most importantly, really, what is your failure plan? So you may have a product that works beautifully, it looks great, and it just doesn't perform as well as you want it to in 1 out of 100 patients. Well, what are you going to do for that one patient where it doesn't work? What's your failure plan? How do you go back in and remove a scaffold from somebody that you've taken out half of their liver, or whatever that scaffold may be? So coming up really with these plans is where the FDA wants to direct your thinking right now in terms of these scaffolds. From our side of things, we obviously want to consider these too, but in the process of trying to create these scaffolding structures, these bioprinting structures, we want to look at things from obviously a big picture and then more of a detail. So big picture point of view is, what's the intended function? So if you're trying to bioprint an ear, your intended function is for it to look like an ear, so from a cosmetic standpoint, and maybe even improve hearing. But some of the secondary functions for that would be, well, does that patient have glasses? Can it withstand the force of the glass pushing down? Do they have kids that are going to come up and pull on mom's ear, reconstructed ear? What are some of the other things that you can think about that the environment for the scaffold is going to be? Again, what's the degradation rate? How long is that going to be there? Or do you purposely want something that will degrade so the body can take over and regenerate its own tissue? Are you going to use it as a carrier for certain cells? So what cells, what growth factors, what do you want to put within this Bioink, like we've heard about, these bioprinted scaffolds within that? How do you implant it? Do you inject it? Do you implant it surgically? Is it something you can do endoscopically? How do you get this into the patients? And then thinking about future modifications. So if you have an implant in there and you need to modify that, what kind of route do you need to do to actually modify that implant? Because now you've put something in somebody's body. And if you need to tweak something or tune something because it's not quite working the way you want it to, how can you gain access to that to modify that? And then also just looking at the details. So how are you going to create this? So again, there's a million different questions and a million different ways to research things, these things. And the FDA wants all of these answers by the time you come to them, which is why it's nice to work with them early on. So creation method, is this something you're directly going to print? Are you going to print a mold that you then shape your scaffolding to? How do you actually create these scaffolds? What type of material? Are you using one material or multiple materials? And if you are using multiple materials, how do those materials then interact? Because materials have very different properties when they're by themselves versus when they're maybe combined with something else. As you're printing, again, extrusion technique, those types of printers are going to print off fiber lengths. So they're going to come off fibers. What is the size of the fiber you want? What are the orientation of the various fibers? Do you want them parallel? Do you want them perpendicular? Do you want them at 30 degrees, 60-degree angles? What fiber angles? Pore size. How big do you want the pores to be? And how about those 3D relation of those pores? If you have a 1 centimeter tissue construct, or does the pore go the entire length of that centimeter, or do you have them offset? So these are all things that, to some degree, you have to have an answer on why you've chosen each one of these things. Why did you choose that fiber size and this pore configuration? And how does that then interact with the body? So you have to go through as just your very foundations to try to get these. Again, to walk you through this process, going back to big picture, what our vision is. So overall, it's to save the world one voice box at a time. It's a very broad thing. But in reality, for this specific thing, is to actually have a larynx replacement that is patient-specific. So if I take a cancer out for somebody, I want a patient-specific scaffold I can put in that perfectly mimics the body's environment. We want something that's there permanently. From the larynx standpoint, we want something to stick around. It can be biointegrated fully and have as much of the native functionality as we possibly can for this stage of our investigation. And then, importantly for us, is that this whole thing needs to be able to be done in about 3 weeks, because I don't want to tell a patient, sorry, you have to wait 3 months. A cancer patient, you have to wait 3 months until we form your scaffold. That just doesn't work. So in order for this to be clinically feasible, it has to be done in about a 3-week time frame. Again, now from the preparation standpoint, again, this is lots of work, and once you get these down, subsequent iterations of these become much easier, but going through the very first process of it. So if you think about taking a CT scan of a patient and then getting that to the point where you can print it, that process also needs to be proved. So, one of the questions the FDA asked us is, what's the minimum resolution on the CT scan that you can use for this process to be valid? I didn't really even think about that. CT scans are just kind of CT scans for me. You know they're good and you can see them. Haven't thought about that. And they want to know what's the minimum resolution that you can use for this. How do you segment? How do you get the specific cartilages and tissues and vocal folds and that type of stuff in the right areas? How do you get the program to recognize those? And then you can take the software and you can change the shapes of those. So if I want to take maybe an ear and I need I'm taking the right side, but it has to have a little bit different shape on the left, how can I modify that in the software? And you can go through that process. And so what the FDA wants to see is, okay, for one patient, I'm going to take that CT scan, and 20 different times I can go from their CT scan to getting that ear to look at exactly the exact same, all 20 of those times, the exact same process. So there's reproducibility in that process. And then can I take 20 different patients and do the same thing for them? So for 20 different patients, can I make something that looks like an ear using the same process? So you can get those two things squared away, then that looks pretty good from, okay, you can reproducibly and reliably do this for patients. Then you send that off to the 3D printer. And the next question, like I mentioned before, is how are you going to actually create that scaffold? Are you going to use a mold, actually create a 3D printed mold that you then shape another scaffold to? Or are you going to directly print that scaffold? And a lot of that has to do with the complexity of the 3D structure. It's a lot harder when tissues are really complex. They have a lot of complex structure like ear to print those directly. Maybe a lot easier just to print a mold and shape another scaffold like hyaluronic acid or something else, like maybe even a biogel that you can form into that shape. So different considerations, again, based off different biomaterials. So here's an example. This is a patient that I have currently that has a cancer within the cricoid cartilage, so within the cartilage of the larynx. And so you can see the process. We can identify where the tumor is. We can see how that interacts with the normal cartilage around that. have an idea of what that defect is going to look like. So the bottom right-hand corner is what the defect would look like if we just simply remove the tumor, not getting any margins or anything. So that gives us an idea now where we're going to have to reconstruct. We can then take that, here's just another view of the tumor in place, and take the normal looking tissue, say, okay, well, we know we want the other good half of that cartilage to look like this, so we can get that, model that, and then take the mirror image of that and print that off, and you can then see, okay, this is what it looked like when we had the implantation in. So basically, it looks like a normal cricoid cartilage without the big tumor there. So how do you get those reconstructed? Again, I'll show you another example of this too coming up quickly. Once you've got all that figured out, now you have to get into the details of the actual scaffolding. So, you know, what we talked about before, fiber size, this happens to be 500 micrometers in size. The orientation, we had to look at parallel, we had to look at 90 degrees, 60 degrees, 30 degrees, 15 degrees. And what does that do to pore size, and how does the cells interact? For this specific scaffold, we found that an angle of 60 degrees tended to work the best. And that's determined through small animal models, how well does that integrate into the tissue, and then a CAM assay, which is a chick chorioallantoic membrane, where you can actually see cells ingrow into that scaffold, and which of these configurations and pore size is that best facilitated. So through that, we can say, okay, well, this orientation has the best strength and the best tissue integration into that. Then you got to add the cells to it. So maybe one thing for it to have good mechanical properties, but how well do the cells grow on that? So here you can see we use adipose-derived stem cells. How do the stem cells integrate with that? And do they actually live when they're on the scaffold? So this is a PCL scaffold. That's what these fibers are. And you can see that we actually get very good growth and survivability of these cells on that scaffold. This is the bioplotter that we use. It's the Vigitech bioplotter. I'm not paid by a bioplotter. I have no relationship other than I probably bought the founder of the company a car by now, I think, with everything we bought from them.
You should get a discount on the next one.
That's right, yeah. That's right. Exactly, yeah. So other than that, I have no financial obligation to a 3D bioplotter. But again, you find the machine that works the best for you and for your needs. And because this is, again, to be into humans, is you have to be able to replicate this. So whatever you're doing in your lab, you have to be able to replicate this in a clean room. So it has to be in a GMP environment. So you have to have two printers. You can't use the same printer that you use for animals for humans. So you have to be able to replicate this. And obviously this is obviously a very expensive process, but it's vetted through the system. This is what our PCL hemilaryngeal graft looks like. Now here it is kind of up close. So that's one thing. So now we have the shape, we have the form, we have the structural, we have an area where the vocal fold is going to go, but that's hard. You know, it's like a table. It's not going to vibrate like a vocal fold is. So then we have to work in there and add a second biomaterial to that area to allow it to kind of get that vibratory capacity of the vocal fold. So you get the vocal folds to actually start vibrating there. So we have to add now a second material, really 3 more materials into the structure. So if you look at the vocal fold, there's different layers within the vocal fold, and we've been able to try to match those layers up with other biomaterials that are very similar to that, using hyaluronic acid, collagen, and some other things to get that to grow. So same thing, you have to go through the entire same process for these separate biomaterials, and we can show that you get an epithelial layer using adipose-derived stem cells and fibroblasts and other cells that actually grow within the construct itself. So we can show that it integrates nicely. And it has the functional properties of normal vocal folds. So obviously our bioprinted construct is in black and white, and the normal vocal folds are there. So you see what's called a mucosal wave. You see that vibratory capability of the vocal folds. So then you can add that into the area where the vocal fold ridge was to get that to function properly. So, and again, all these separate steps we talked about need to be documented throughout and to be able to show reproducibility of that. So this is what the vision really looks like when all is said and done. Is you have a cancer patient with a laryngeal cancer. They're going to come and see you, and typically the next step would be to go and get a CT scan. That helps us obviously in our operative planning, but then just like we had talked about from there, you can take that CT scan and then you can modify that. So here we have the 3D printed, or sorry, a 3D diagram of that, remove what we would have for the surgery, take the good side, make a mirror image, and then modify that using our software. Again, proving all the way through that we can reproduce this over and over and over again for people. You print off your scaffold either directly or through a mold. You can add secondary scaffolds, which is kind of represented by this kind of orangish thing, use it to help growth, whatever it is you need to do with that. While you're in the process of doing that, we just get fat from the patient using liposuction, and you guys know this process pretty well. You can separate the adipose-derived stem cells from the fat cells, expand them, differentiate them in this bioreactor, circulating bioreactor, what people use for tracheas. That process takes about 3 weeks. It's not fully epithelialized there, but it has a base layer on it. And then we can show then through our animal models that the actual native stem cells within the body will come in and help to epithelialize that segment. So in about that 3-week time span that we shot for, we can show that we can at least get a seeded scaffold that we can implant at the time of surgery. So that whole process has kind of gotten us to where we can now get these into patients to start working slowly through this. And then we still don't know, obviously, the outcomes from that. But I think one of the key things and one of the take-home points that was really big for us as our program develops is getting in and working with the FDA early. They're more than happy to get in and work with you, saying, here's our end goal. This is where we're beginning, and this is what we've done to now. Where do we need to go? Because I think the trap that a lot of us fall into is we go down the road, then we talk to the FDA, and they said, well, no, you did that wrong. You had to go back and do it. So you end up kind of going back and redoing things over and over again. And in terms of production, that's not obviously the best way to get things safely and quickly into patients. So thank you very much. I think we're going to have a discussion now. Thank you.
Great.
So I think we'd love to open up to questions. I'm sure we've got quite a few. Anybody with specific questions, you can direct it to the panel in general or individual members of the panel. Otherwise, Josh and I will open up with some questions for our panelists, and you guys can ask questions too. Yes? Sorry.
When you were talking about your organoids and how you were using them for toxicity testing of drugs, how do you relate the actual result you get from your organoids?
I just wondered how you related the toxicity that you get from your organoids to that which you find in patients. I mean, if you find like one drug is toxic at 1 micromolar, what does that mean for a patient?
Well, the key thing you have to do, and what unfortunately, it's unfortunate and then maybe in the future fortunate, you know, with a commercial plan that Organovo had to build and understand how to market something like this. What was unfortunate about it is that the ante to come to the table for pharma, for them to really kind of consider, you know, whether they would use your platform or not, was to test at least 30 drugs and have a broad set. So within that you would have known toxic drugs, you would have known not toxic drugs, you'd have drugs that are on the border, different mechanisms of toxicity. You had to have a blend of things, and then looking at that constellation of results, you can compare. And importantly, you're also identifying not really biomarkers, but you're identifying the markers in your system, because they don't always exactly match up, right? Are you going to use ALT elevation specifically, or are you going to use albumin drop-off, because that's showing the death of the cells? What is exactly predicting toxicity in each case? And so it's you get a set of results, and then you have these markers you can rely on. And then the reason I say that's somewhat fortunate is that then if you kind of start to build credibility around that and some momentum, well, then anybody else has to pay that, you know, 7-figure ante too to come to the table and really start to look at these things. So that's the biggest thing. There's a whole set of drugs that have been tested, and you really do it by this constellation of results. It's not one result. But I would say within that, what Organovo has been able to do is show that for most, not all drugs, but for most classes of drugs with specific exceptions, the comparability, one of the nice aspects of the platform is that you do see dose relationships. So it's not the math is not exact here, meaning that when you expose a drug in an in vitro context, it's a constant for a period of time, versus in the body where a Cmax is a peak. But if you expose Organovo's tissues at a Cmax, that tends to replicate the result that you see in the human at that Cmax, But of course, the Cmax is a profile. It's part of a profile in humans, and it's not the constant. But then, but if you use that relationship, and they do this all the time when they compare animal tox results, they're comparing the relationship. It's 1-to-1 for most drugs, which is nice. Versus certainly in a 2D setting, you know, the results that you get are at 30x or something like that. You have to give 30 times the toxic dose of a drug to see the same result in a 2D tox setting because those cells aren't performing normally.
So along those lines, while we're on the organoid models, what about genetic diversity and capturing that? And maybe, Tom, you could also speak to this as well. And then in addition to that, I was wondering if you could speculate on how long is it going to be till we're using these models and replacing the animal models in the regulatory process? Like, are we talking 5 years? So the
in the regulatory
diversity
process for what use? For everything or for some?
For pharma.
Okay.
So pharma would come and they'd say, rather than do these host of animal studies, we're now going to develop these organoid systems for looking at toxicity, for instance.
Yeah. So one helpful data point, and this was one of the reasons I used Vertex as a learning tool, as a case study, is that Vertex's cystic fibrosis platform, which is multiple drugs launched now, was built entirely on a 3D cell culture. They never did animal pharmacology with those drugs that they launched, meaning they relied entirely upon in vitro 3D cell culture models that they had developed internally for the efficacy, for the pharmacology result of the drug. They still did normal animal toxicity. So that's a good case that it can be done already. FDA will listen to a holistic scientific argument. If you can't replicate it in an animal model, cystic fibrosis in this case, it's not going to be helpful to get patients drugs anyway. But as to when it'll be used more broadly, everyone has an opinion. I'm sure mine is. Pharma is a highly inertial business, right? So I think it takes a long time. Decade.
Question here.
So when I look at the future of tissue organoids in generating preclinical data for FDA applications, I see it as sort of sitting in between a large animal model and all your small animal models and culture studies. The systems are inherently variable based on your cell sourcing. We get different performance out of our organoids depending on who, where we're buying the cells from to put into it, and it's not cheap. And in order to set up, at least with us, with our integrated systems, where you've got 6 different tissue organoid types, each of them have between 3 and 5 different cell types in them. They have to be matured individually. They have to be conditioned together so that they survive and function under a common media. I think that the proof will be in the pudding if we can continually demonstrate, as we have with triglycerin and estemazole and other FDA withdrawn drugs, we've been able to pick up toxicity, and when we do the math, it looks like we're picking up toxicity at concentrations that are similar to the serum concentrations of patients that were taking these drugs. If we can show them that these complicated, expensive, and variable systems that have flaws are given them a heads up that might save millions, if not tens and hundreds of millions of dollars, I think that they'll raise an eyebrow eventually. And if you had 2 drug candidates and you were considering pushing those forward, wouldn't you like to know that one of them, hey, we started to see some cardiac arrhythmias in our cardiac organoids with human cells? It's a piece of information I would imagine that a drug company would like to have in their back pocket when they are making decisions and long-term plans as far as their candidates go.
Thanks, Tom.
So we have a question out in the audience. Yeah.
Thank you. I wanted to ask, for toxicity testing, what do you see are the pros and cons of 3D printing versus Organoids-on-a-Chip technology? So it'd be interesting to see what the panel thinks about that. Some of the organ-on-a-chip companies promote angiogenesis as a differentiator that they have, but interested in your views.
Well, you take a crack at it and I'll follow you.
Okay, that's fine. So maybe let's actually say there's maybe 3 levels here, because it would be a tissue organoid like an Organovo that I described, a spheroid as a step, and then organ-on-a-chip, which can be, it can be multiple things, right? So the microfluidics aspect can either be with a 2D cell culture, or it can have some 3D component or a spheroid in there. So that can vary. So I'm not familiar maybe with the specific example you're giving in terms of claiming angiogenesis as a benefit. That can be a benefit. I think you need, it's really along those lines of like how much throughput you get and how high content is the result. What you're talking about is that trade-off, I think. And I think you can add value over what has been done before at every level, and you don't know where you get the max sweet spot. It may be, in some cases, that a highly complex tissue is overkill because you just needed a little bit of more complexity to get certain functionality. So for example, something that is 2.5D, if you want to say it, or not even, certainly not 3D, if you think about it as Heparin, which has this I don't know what they're called anymore. They've changed so many times. I'm sorry, I don't know the current name. But Heparin, if people are familiar, has this micro-patterned 2D culture where they've got fibroblasts surrounding islands of hepatocytes. And they see that in predicting metabolism of the drug, so how it breaks down, what it breaks down into, what metabolites it breaks down into, they're really good. They're much better than what was out there beforehand. But then when you go to the next level in toxicity, maybe they're not as predictive. So you can get some value at every step, I would say. And then spheroids are going to add more value, certainly. Do you need to go more than the spheroid? Maybe you need exact control of the structure within a spheroid. And maybe you can get that with a bioprinted tissue, or maybe not. So there's these levels, I think, and I don't know. I think it's going to depend on the disease area or tissue area and the exact application, what the best thing is. So maybe just to say one more thing. So organ-on-a-chip that I'm familiar with in predicting toxicity, there's like a UCF, I think it is, professor who does work where he's demonstrated, for example, that you can, and you guys, I think, have done things like this too, where you can take a pure 2D microfluidics system where you've got liver and then kidneys and then cardiac cells separately in separate chambers, and you can show that the drug, a specific drug, will be metabolized by the liver cells and then result in metabolites that are toxic to the cardiac cells that you wouldn't see if you just exposed the starting drug to those cells. And that's a level of complexity, but would it do every drug the same way? Probably not, because those are 2D hepatocytes, if you see what I mean.
With the in terms of the bioprinted constructs, obviously Keith showed you that you can keep human hepatocytes alive and functional for a sufficient amount of time to do testing, and that's more high throughput than what we do, but that's an accomplishment. Another example of the 2D versus 3D, we were doing some experiments with propanolol and epinephrine and blocking the effects of epinephrine with the beta blocker propanolol, having the liver metabolize that propanolol and recovering the effects of the epinephrine. And we were trying to get some control media with metabolized propanolol in it to set up some mass spec that we were doing for metabolites. And we thought out an entire vial of human primary hepatocytes, basically with 2 million cells. We were unable to get any metabolism of the propanolol in 2D, and we got all the propanolol metabolized with 50,000 hepatocytes in spheroids, and over the same period of time. So the level of functionality in 2D versus 3D is incredible. And as Keith said, with the tissue organoid system, with the integrated organoids, and now we have multiple 6 organoid setups that we've run and kept alive for a month with no loss in viability or function. And we were doing an experiment, and both these experiments I'm talking about, we'll talk about in the next session. we were doing a bleomycin-induced injury in a lung organoid, and we were basically looking for IL-8 coming off the lung, showing that we were causing inflammation in the lung. But our cardiac organoids stopped beating, and we didn't know why. And you know, when we added the bleomycin directly to the cardiac organoids, they kept beating just fine. And we looked into it a little bit, and one of the things we noticed that was that the inflamed lung organoids were producing high levels of IL-1 beta. And we dug through the literature, and sure enough, we found that IL-1 beta can affect the function of cardiac tissue. So then we bought some IL-1 beta and added that directly, and sure enough, that stopped the heart from beating. So, you know, here we were looking at a study where we were expecting to see something in the lung, something happened in the cardiac, and we were able to go back and trace a route where the primary injured organoid sent off a signal that affected the function of a secondary organoid. And those are the kind of things that make us excited that, you know, maybe you don't have to know specifically what you're looking for in these systems, because you know, we can monitor the health and the viability of all the cells in the system, and if something happens, well, it may be a red herring, but it's probably worth tracing backwards and figuring out what happened.
Other questions? I have one for Dr. Lott. So one question I would ask is, first, maybe just to set the table, is the goal of your overall effort, is it to treat patients that come to you guys under the leadership of a clinician at your center, or is it to kind of build to a product that's widely distributed?
I guess a little both would be the best answer to that. So to me, the goal of any research that you do is to always have it widely applicable. So in trying to figure out that question, you know, it's obviously that whole process is very difficult to do, but is there maybe something within that process that you could then apply and say, okay, well, we can create these scaffolds and have those ready for patients, and then have someone locally do the stem cell work, and here's the recipe to do that type of thing. So I don't know how in total the entire process would work at, you know, maybe community hospitals or things down the road. But once we can prove that that process works, then what parts of those can we then maybe give to external places to make that easier for them?
And the reason I ask is, and your answer takes you in one direction that I think you need to go in. I think you were talking a lot about the FDA and your work with them, and I think that's the appropriate path. But as Peter Marks, the CBER director, said yesterday, you know, everything is regulated for them. All the things like more complex tissues, as opposed to graft tissues, are regulated under 351. And as someone on the panel pointed out, that is an interstate commerce clause regulated act, meaning that it only applies if you are intending to introduce your product into interstate commerce. So there's a ton of clinicians, and some of them were at a session at 11 o'clock this morning doing work, clinicians doing work, as opposed to companies trying to make a lot of money to help their patients. And so the stuff that is done with the stromal vascular fraction or the adipose, to treat breast reconstructive surgery. Things like this are all done, and the FDA is not interacting at all with those folks, to my knowledge. But I think that the dividing line for the FDA is, are you seeking to help individual patients who are coming to you, or are you seeking to make a big enterprise, make a lot of money, and do advertisements on in-flight magazines and things like that? And that's where they're starting to take enforcement action, is when your intentions are not so good. So, but for some efforts, I think you could go a different route. And then I had a follow-up. Sorry.
Sure,
So I think we got a few minutes, or 1 minute, I guess. There's the tracheal work. Sorry to ask you about this, but that tracheal work with Paolo Macchiarini. And my takeaway, although there's the very negative aspect that he didn't do the right things from a regulatory perspective, right? But that's not what I'm actually asking about. I'm saying, let's say they did all the right things from a regulatory perspective, got similar experimental results. My takeaway from that was that he used human scaffolds in the beginning, so cadaveric scaffolds in the beginning. He switched to a PCL-based scaffold from University College London, and that's when they started to have significant challenges. So are you concerned at all about that, and how are you sort of trying to think about individualizing these scaffolds like you talked about?
Yeah, no, that's a great question. And that's a great example of making sure all of your work is obviously clear up front, you know, kind of that see-through kind of mentality. So a couple things to answer that. So when you look at decellularizing tissue, especially for the larynx, you do run into some issues with that because it's not patient-specific. So you'd have to find the exact perfect match for a specific person. So for the larynx, it's a little more difficult to do. For the trachea part of it, it does make a lot of sense, but it's very hard to get those osteocytes out of the cartilage. We've done that before too, and we did a study using pigs and had very difficult issues getting those last kind of few residual, although you still didn't mount a big immune response with them. So one of the big reasons why we switched to the larynx is, I think in research, your model dictates a lot. And so when we look at people with larynx issues, one of the nice fail-safes in there, we talked about what do you do in the case of failure, is these are patients that we're going to get a laryngectomy. And the only option really for them at this point is to have their entire voice box taken out. So if we put in a half of a larynx or a portion of a larynx, and they have problems with cannulation like Macchiarini's patients did, have those issues, then the fail-safe is they're going to get the same surgery they would have otherwise. So it's a lot easier to me ethically and to talk to other people from a regulation standpoint that, hey, our fail-safe here is still okay. The patients are going to still be okay when they're done. In the example that Macchiarini was, when your trachea is gone and you have nothing else to reconstruct, you're in big trouble. There's nothing else to put back in there. And again, these were people, in fairness to him, these were people who were likely, some of them, going to die. Some of them, he fudged the truth a little bit on that, a lot. And they weren't going to die. So that's where kind of the example of, you know, compassionate care comes in. Are these truly compassionate care, or are you doing it for personal gain?
Yeah.
Yeah, so I guess to me, that clarity, and then have the one last thing that we're doing, I think is a little bit separate, I think it's important to do, is as investigators, we're all very much invested in what we're doing. So all of our trials, I actually have an external board that has a patient member on it. So that kind of reviews what we do. So if we get halfway down a clinical trial and we say, hey, you know what, we're doing these larynx things, we're having a problem. And for me, because I have so much time and money invested in it, I want to try to push it. Well, everybody else may say, hey, well, you know, realistically, this is not the right route to go down. And I think having a patient representative on that is a big, big improvement.
Thank you. All right, with that, I think, any comments, Joshua?
No.
I think we have to thank everyone. Our time is up, but thank you so much for your attention today, and appreciate it. A hand for the presenters today, please.
Thank you, guys.
Great. Thank you all.